So I'm just opening my whiteboard. So today I will be starting with evolution, Hardy-Weinberg principle. So I'll share my whiteboard. Just tell me if it is visible or not. Is the whiteboard visible?
Yes, sir.
Yes, sir.
Okay. So the first thing which we will be talking today is on Hardy Weinberg law. So essentially, this is a law on population genetics. So what you study over here is actually the genetics, you can say inheritance on the level of a population. So now we have to understand what is a population. Population, or specifically in terms Mendelian population. This definition is theoretical. Practically, it does not occur. So, what it says, it is a group of actual or potential. They have the potential, but they don't do. Actual or potential interbreeding individuals of a species. So they breed, or they have the potential to interbreed. So if you consider in a population, there are males and then there are females. So as per Mendel, a Mendelian population, all the males and all the females, they have the capacity to interbreed among themselves. Theoretically, they don't do. Think you don't breed among your siblings or among the parents. You don't do it normally. But in plants and all, you do. They do it. They cross with the parental. They cross with the offspring. They do it in plants and in Drosophila. It is done actually to study the inheritance. But in reality, this is a potential. They have the potential, but they don't do. It doesn't take into account. Just 1 second, I'll just put this up. This is disturbing a lot. এটাকে একটু পুছে দিয়ে হিজিবিজিটা এসে গেছে। Okay, so actually in reality, this does not take place. There could be the many factors. Same siblings, attraction, society, that includes the religion. Several of these factors, they don't allow this theoretical thing to take place, potential interbreeding individuals. So, the lowest level of a Mendelian population একদম ছোট করে দেওয়া হচ্ছে, it is a panmictic population where every male, where all male mate with all female, and this is done random. This is done in a random manner. So, one important aspect which we talk over here is random mating. So, essentially, now to show the relationship among species, population, and individuals. Now consider this to be the whole big circle. This is species. Now in this whole big circle, there are several, among the species, there are several populations. Random mating, and a few aspects of population you have to remember from ecology. There is a time component at a specific time. At a specific location. They must be of the same species. So they are individual populations. And in between the population, in population, you have these green circles. They are individuals of the population of the same species. They belong to the same species. And so these greens, they are essentially individuals. Now let us go to the next aspect. So Population genetics essentially it studies inheritance at the level of population, not individual. They are least bothered of one individual has a mutation or not, but they are more bothered in how many individuals in that population have that mutation. So basically, what we know, in a particular population, there are people, individuals which have got genes in a population. So a gene is represented by, in an individual, it has got a maternal component and a paternal component. So this is one N and this is another N, and together they form 2N. Now, a particular gene, it has got alleles. So what are alleles? Alleles are the contrasting forms of a gene. A gene for height. You can have the tall form or the dwarf form. So this is one allele and this is one allele. So T and T, they are the alleles. So the alleles can be, say, capital A, small a. They are contrasting forms and they are alleles. There also can be something known as multiple alleles, like in our blood group A, B, O. So the gene is essentially, gene for blood group is I. So you have a maternal component. If the maternal component, the alleles are, it can be IA, the contrasting forms, the alleles IB and only I. This is O essentially. So these are the alleles. So individuals who are IA, they show the blood group A. Individuals who are IAIO, they are also blood group O. Blood group A, sorry. So this IAIA or IAIO, they are known as genotype. And this blood group A, which is present, is known as the phenotype. They can also be the allele IB, IB. They will be blood group B. Then they can also be IB, IO. This is also blood group B. This is the genotype as usual. You could have blood group IA, IB. They so this actually follows a Mendelian principle known as the law of dominance. We are talking about individuals at the moment. We are not gone to the population. That is, IA is dominant over IO, and here IB is dominant over IO. So IO is not expressed. When it is IA, IB, these individuals have got a phenotype of blood group A and B, both are expressed. This means IA is equal to IB, and this phenomenon is codominance. It is an extension of Mendelism, phenomenon called codominance. Apart from that, this is the genotype and this is the phenotype. The next option which you get is IO. There's some people write II also. The genotype, these individuals blood group O. So the allelic series will be IA equal to IB, which is dominant, or both of them, they are dominant over IO. This is on the individual count. Now we come to the level of population. We have understood what alleles are, and now we come to what a genotype is and what a genotype allele is and what is a genotype is. Now, in individual level, we have understood what allele is and what a genotype is. Now, when we consider when we are talking in terms of population, in terms of population, you are concerned with what proportion of the individuals have got blood group A, what proportion of the individuals have what proportion of population has blood group A or B or O. This is one question you are interested. The first question. The second question which you will be interested is what proportion of the population has allele for IA or IB or IO, for example. This is what you may possibly be interested in. So this brings into the question, the first question, that brings in a term known as genotypic frequency. What is the frequency of individuals which have got IO genotype? This is the question. And the second question which comes is, what is the proportion of individual with IA? And that is known as allelic frequency. Now let us go to a theoretical model. So now there is a term known as gene pool. So in the whole population, what is the proportion you have alleles IA going on? Or let us consider a theoretical. There are two alleles, capital A and small a. So you have individuals AA, 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 AA. Like this, you have lots of these all these individuals moving in the population. So the gene pool is the sum of all the alleles in the population. That is a gene pool. So properly, if we say the sum total of all different alleles in the breeding members, let's write it properly. Gene pool, it is the sum total of all the alleles, rather all the different alleles in the breeding members of a Mendelian population where random mating takes place. Population at a given time. So this is the gene pool. And now the genotypic frequency can be defined as it will be defined as the percentage of different genotypes found within a population, and the allelic frequency will be proportion of different alleles of a gene in a population. Now, technically, let us calculate. Let us go to the question calculating genotypic frequency and allelic frequency. So, it is calculated on the basis of a locus. So, in a particular for a particular gene, there are two loci, one the maternal and one the paternal. Two loci. One loci can have this, the other loci can have this, or it can say in the population which has got two alleles, capital A and small a. One loci can have this, and the other loci can have another genotype. So these are the essentially the for a population which has got capital A and small a as the two alleles, three possible genotypes are present. Now, the three genotypes which are present, imagine total population is 1,000, and in that 1,000 people, we know that AA individuals are 450. The heterozygous population are say 400, and small a people are number in numbers 150. So total is 1,000. Now you are trying to calculate what is the genotypic frequency of AA. or you can say frequency of AA individuals. It is very simple. 450 people are present divided by total 1000 people. You can call them to be 0.45. What is the frequency of people which is like this? You can calculate again. 400 by 1000, 0.40. And frequency of small a. You can calculate 150 divided by 1000. You will get 0.15. Is this clear? বুঝতে পেরেছো এত অব্দি?
Yes, sir.
হ্যাঁ স্যার।
So you have found out the genotypic frequency. And so the total frequency is plus frequency of AA plus frequency of AA. This is equal to 0.45 plus 0.40 plus 0.15. This is equal to 1.0 or 1. This is the maximum. So total frequency of the whole population is 1. Now, the allele frequency can be calculated by two methods. The first is, this is known as the gene counting method. So allele frequency, the formula is number of one and an allele. It is the number of an allele. I'm underlining, of one allele or one type of allele. There are two loci. So therefore, at a given loci divided by total number of alleles in that loci. So we know that AA individuals have two alleles of A. AA individuals have got one allele of A. Similarly, AA individuals have got two alleles of small a, and AA individuals have got one one allele of A. So from this, the formula can be derived into allelic frequency is 2 into, so A, let's use another color. So number of A calculate, you can take this. So this is 2 into and this is 1 into. Okay, so the formula will be for allelic frequency 2 into count in homozygotes plus, this is 2 into count of homozygotes plus count of heterozygotes. Two into divided by number of, we are just number of alleles at a given leka divided by 2 into total number of 2 into total number of individuals, because there are two loci,
স্যার
therefore 2
লোসাই মানে কি?
loci হলো একটা পয়েন্ট, একটা ম্যাটারনাল কম্পোনেন্ট মার থেকে পায়, একটা বাবার থেকে পায়, ঠিক আছে, মাদার থেকে পায়। তো এই লোসাই হচ্ছে যে পয়েন্টেতে জিনটা থাকে।
আচ্ছা ঠিক আছে।
লোসাই ইজ দ্য ফিজিক্যাল লোকেশন অফ দ্য জিন বলো বা অ্যালিল অন দ্য ক্রোমোজোম। ক্লিয়ার?
ইয়েস
ঢং
স্যার।
করে আওয়াজ, ঢং করে আওয়াজ হলো কেন?
হ্যাঁ, হট করে লেফট হয়ে গেছে স্যার, আমারটা আবার জয়েন হয়ে গেলাম এই জন্য।
আচ্ছা আচ্ছা, ঠিক আছে। Loci is the physical location of the gene on the chromosome. তাহলে এই ভিত্তিতে যদি আমরা ফলো করি, what is the, এবারে বের করছি allelic frequency of A, 2 into count of homozygotes, তাহলে 2 into 450, 450 জন ক্যাপিটাল এ ওয়ালা রয়েছে, তাহলে কতগুলো তাদের থেকে আমরা অ্যালিল পেতে পারি? 2 into 450, ক্লিয়ার?
হ্যাঁ স্যার।
ওকে, plus count of heterozygous. How many people are heterozygous? 400 of them are heterozygous. Capital A, small a ache, they have. So 400. So how many total? 1000 individuals. So how many loci are present? 1000 into 2. One from father, one from mother. Into 1000. This is 0.65. So the allelic frequency of capital A is 0.65. Okay.
ওকে স্যার।
এবারে similarly you can calculate this in a similar manner, 2 into 150 plus 400 divided by 2 into 1000. This is 0.35. You can also calculate 1 minus, so frequency of A is equal to this. তার মানে you can also do it as 1 minus 0.65, you will also get 0.35. This is an alternate way you could have done. Instead of calculating this, you could have done it like this also. Now, the second method is based on genotypic second method is based on genotypic frequency. Suppose you are only given genotypic frequencies. Total number of population or how many people are there of capital A, capital A, it is not given. Only the frequency is given. Then you are told to calculate the allelic frequency. So you are only given, say for example, frequency of AA is 0.45. Frequency of AA is equal to 0.40 and frequency of AA is 0.15. You're only given this. That's all. Now your thing is to calculate the allelic frequency. So there is a formula for this, and that is frequency of a given allele equal to frequency of homozygotes plus half frequency of heterozygotes. So from this, you can calculate frequency of A is equal to frequency of homozygotes, frequency of AA plus half into frequency of AA. So in this case, it is 0.45 plus half into 0.4. If you calculate this, 0.2, this will be 0.45 plus 0.2, तो दो बार जाए 0.65, and frequency of AA, you can calculate frequency of half into frequency of AA, 0.35. This also can be done as 1 into. Clear? So far clear?
Yes,
No?
sir.
Okay.
Yes,
sir.
sir, clear.
So today we stop over here. Day after tomorrow at 6 p.m. or 4 p.m. I am bole debo. We will continue with the second part of Hardy. We have not yet started the Hardy-Weinberg principle, but this introduction is very necessary before we go to the Hardy-Weinberg principle. So allelic frequency, gene frequency দিলে তোমরা তো ইয়ে পারবে না, করতে পারবে তো এই অঙ্কগুলো?
হ্যাঁ স্যার করতে পারবো।
Now এরপরে একটা another, there is a Hardy-Weinberg principle. You will see another way of another type of calculation is, this is the basic how to understand calculate the genotypic frequency and the allelic frequency. এটা একটা part of the bigger problem. ঠিক আছে?
ঠিক আছে স্যার।
কোনো কোয়েশ্চেন
Okay, sir.
আছে?
না স্যার।
না,
ওকে,
sir.
দেন আজকে শেষ করছি।
ওকে স্যার।
পরশুদিন আমি লিংকটা দিয়ে দেবো তোমাদের।
ওকে স্যার। থ্যাংক ইউ স্যার।
ওকে স্যার।